Okay, so I'd first like to thank the organizers for this really wonderful meeting. It's been a great 2 days, especially Venki for initiating this, Joel for putting up with me, and Richard for making sure that there's somebody still in the audience to listen to my talk. Okay, so every time you take a breath, every time you have a meal, your very important and sensitive internal organs, of which there's about 200 square meters of exposed epithelium in your lung and guts. They're exposed to biological and physical hazards that could be life-threatening if we didn't have something to protect us. And that something that we have to protect us is mucus. Now, I'm going to be talking about mucus today, starting with the gut, with the intestinal mucus. But many of you are familiar with mucus from another setting, and that's this one here. Okay, so what I'm going to do in this talk is we're going to get a little bit mechanistic. I do want to discuss a very complicated macromolecular system. I'm going to not talk about my favorite arginine, but I will talk about my favorite disulfide bonds. So you're going to have to pay attention every once in a while if you want to follow the mechanism. But if you don't, that's okay, because what I want to do in the big picture is just to bring us back, those of you who are not physicists, to bring us back to the time when we decided to be biologists and biochemists. And I don't mean in high school, I mean when you were 5 years old and you just wanted to know how really disgusting things work, okay? So mucus is disgusting, right, but it's also wonderful, and we can have some debates about what exactly is the fabric of life, but I'd like to make the argument for the next 45 minutes that mucus is in fact the fabric of life. Now, as I said, its job is to protect the exposed epithelium. And mucus is produced by these mucus-secreting cells called goblet cells. Here they're shown in the respiratory tract, but as you'll see, they're also in the intestinal tract. And in the respiratory tract, the epithelium has cilia, and the mucus is secreted onto the epithelial surface. These cilia they help to roll the mucus along up our respiratory tracts, clearing out those pathogens, clearing out the particulate matter that enters with the air, and bringing it up. Okay, now most of the time we don't see the mucus, right? That's not our typical situation unless you have one of these little cute things at home. Most of the time it just disappears. Now, where does it disappear to when it's coming out of our respiratory system? Well, it just goes down into our intestinal system. And you may be interested to know that about 1.5 liters of mucus every single day is coming up out of our respiratory tract, cleaning it out, and going into our GI tract. So for those of you who remember that kid in first grade that was sitting there picking his nose and eating it, or for those of you who were that kid in first grade who was picking your nose and eating it, don't worry about it. It was a drop in the bucket. Okay. All right. So as I said, mucus is present not only in the respiratory tract, but it's also in the GI tract. And here we have intestinal villi. All these white dots that you can see on the surface are these incredibly large sort of multivesicular structures called goblet cell granules. Oh, thanks. And about 10 microns across. This is incredibly densely packed mucus, but when it's released into the environment, it's a hydrogel. It expands enormously, about 1,000-fold, absorbing water. The change in pH, the change in ion concentration, is what's responsible for this phase shift. Okay, so the proteins behind mucus are illustrated here in their domain diagrams. So MUC2 is the intestinal mucin, and 5AC and 5B are the lung mucins. And we're going to focus on the intestinal mucin for the first part of this talk. So there's some domains at the beginning, there's some domains at the end, which are important, but in the middle is a region of prolines, threonines, and serines. That's why it's called PTS here. And I'm sorry to Sarel and Joel, but acetylcholinesterase is not a monster protein. This is the number of amino acids within these mucins. So this is 10 times bigger than acetylcholinesterase. This region here is 2,500 amino acids, heavily decorated with O-glycans on the threonines and serines, such that 80% of this molecule by mass is actually carbohydrate. Okay, Gideon, you heard that? Okay, so you cannot run these proteins on a gel. Not even an agarose gel, and this is something that brings a biochemist to tears. Okay, it has brought our lab to tears many times. So this is just an illustration of what this PTS region looks like. As you can see, it's highly repetitive. All right, so mainly I'll be talking about mucins in higher organisms, but mucins have been around a long time, and they basically originated with the earliest multicellular life forms so corals, comb jellies. And these organisms are very simple. They're basically all epithelium with a little bit of jelly-like layer in between. And protecting both the outer and the inner surfaces of these organisms are the first primitive mucins. So it is, in a sense, a fabric because these organisms are wearing their mucins in a sense that we might not think that we're wearing our mucins. Interestingly enough, just as an aside, marine biologists discovered only 3 years ago that these organisms were previously thought to eat and then just to spit back out wherever they couldn't digest. But only 3 years ago, it was discovered that they actually have a butt. So they actually have a gastrointestinal tract that goes all the way through, and they poop out what they don't need at the other end. All right, so mucins evolved early in multicellular life, and they're fantastic molecules because they're great for elasticity. They have a tunable rheology based on the ions and the pH, and the ability to self-repair. So they're very important for protecting the organisms physically. But in addition to with the evolution of mucin, we also evolved the ability to distinguish between the microbiota in our environment and the microbiota that we want associated with us. So for example, sponges, right, which don't have mucus, just indiscriminately have all the bacteria and microbes in their environment stuck to them. Okay, they're just sponges for the microbiome in the area. But corals and comb jellies, which do have mucins, can actually distinguish between microbes that they want to be with them and microbes that they don't want to be on their cell surfaces. Okay, but let's go back to higher organisms. This is what the mucus coating looks like in our intestines. So these are the mucus-secreting cells, and on the surface of the epithelium, there's a very tight or very densely stacked layers of mucins, and stained in red here are bacteria. Okay, so this is our microbiome that's existing in our gut, but as you can see, it's held apart from our cell surface, from our epithelial layer, by this densely packed mucins, and only when there's a phase of more loosely packed mucins can the bacteria actually get into the mucin layer, live there, they eat the mucins, and they then compete with the pathogenic bacteria that we don't want to colonize us. Okay? So that's what the mucins look like in our gut. Now, I didn't start off studying mucins because I wanted to study mucins. Of course, we got there through the main interest in my lab, which is disulfide cross-linking. And I've been fascinated by cysteines and disulfide bonds since graduate school. They're strong but reversible bonds. Cysteine is an incredibly versatile chemical group, both for biochemists, for labeling, and also to carry out biological reactions. I'm going to argue that in the future, cysteines are going to be a very important control point for tissue engineering. So it's worthwhile understanding cysteines, understanding disulfide bonds in biological systems. And through work that more biologically oriented that I won't tell you about today, we ended up focusing more closely on mucin structure and function. So this is a very small part of this 5,000 plus sequence of mucins. This is not the threonine serine rich portion. This is one of the parts at the N terminus. And as you can see, it's just full of cysteines. So this was a big challenge for us to understand how these cysteines are forming disulfides and how that contributes to the structure and the properties of mucins. So when we first encountered mucins in the literature, this was the story that was kind of told to us. So as I said, mucins are very, very long proteins. They have a bit at the beginning, cysteine-rich bit at the beginning, cysteine-rich bit at the end, and a long stretch of carbohydrate-rich portion in the middle. Now, these proteins, in addition to being enormous, they become more enormous by forming disulfide-bonded dimers in the endoplasmic reticulum by their C-terminal ends. And then these dimers were thought to further polymerize, to trimerize via their amino terminal ends, the white end here, making that kind of honeycomb-like lattice. And if you look at if you guess the dimensions from the size of the edges here, it would basically make mucins serve as a 0.2 micron filter. So perfectly suitable for filtering out those bacteria, preventing them from getting to our epithelial cells. And if you imagine taking layers and layers and layers of this substance here and just turning it 90 degrees and stacking them up one on top of another, you would get something that looks like that and performs the job of filtering out bacteria, the microbiome, and keeping them away from the cells. Okay, for various reasons, we wanted to explore in more detail the biophysical mechanism of mucin assembly and to try to understand at cysteine residue resolution how these proteins actually come together to form these larger structures. So we did this by focusing on the beginning part of the of the mucin protein. So this is the part that's supposed to undergo this disulfide-mediated trimerization in the late secretory pathway. And we produced this part by itself and began to study it biochemically. And the first thing that we discovered is that this beginning part of the intestinal mucin protein is actually not the trimer that we expected from the literature, but is in fact a very nice dimer. And if you produce it either with this little orange part and gray part at the end or without, in both cases it comes out as a dimer and not a trimer. So that led us to suspect that this trimerization model, the hexagonal lattice model, was not actually what's happening. It was a beautiful, elegant model that explains some things in biology, but it probably isn't correct. And instead we were thinking that, in fact, the mucin molecules undergo disulfide-mediated dimerization in the Golgi, and they make these long polymers. Now, that's unfortunate for this beautiful model that was actually useful because it explained some things in the gut, but it's actually better for unification of biology that we have a dimerization situation in gut mucins because lung mucins also form dimers, and they're thought to form long polymers here. And the homology is so strong between the gut and the lung that there's no real reason to expect them to do something different one from the other. Okay, so that's great, right? So now we have the idea that we have a unification of the lung and gut mucins. We have disulfide bonded chains of polymers that are making up this fantastic structure. But we come back to a fundamental question that's actually been present in the study of these molecules and related molecules that I'll explain shortly, which is if you have the ability to dimerize by the N-terminus, the ability to dimerize by the C-terminus, and a lot of flexible P and T and S-rich stuff in the middle, then how, in fact, do you form a long polymer of this protein rather than a closed dimer? So this was the puzzle that we were then faced with, right? How do you make sure that you only dimerize molecules by the C-terminal end that are not dimerized by their N-terminal end? Okay, so just to orient you, the structure here at the carboxy terminus, which is supposed to make disulfide bonded dimers in the endoplasmic reticulum, was determined, at least a homologue of it, was determined by Tim Springer's lab at Harvard Medical School a number of years ago. So this little domain that you see here represents this species here, which has 3 intermolecular disulfide bonds. But how this structure looked, the beginning, the N-terminal region, and how it accomplished this task rather than this task was unknown. Okay, so I mentioned that, the N-terminal region of mucins forms dimers if you just look at them by secmols at neutral pH. But if you put them at low pH, they do something very interesting. And that's that they polymerize into these structures shown here. So they make a beaded polymer at low pH. Now, when we saw this, by negative stain EM, Gabe Javits, who's a fantastic direct PhD student in the laboratory, said, I want to solve the high-resolution structure of this. And I thought, okay, well, try to get it to be monomeric so that you can have single particles. And together with Nadav Elad, who's here in the audience, who's our single particle expert at the Weizmann Institute, they said, why, right? It looks great as polymers. Let's just solve the single particle structure of the beads in the polymer. And that's, in fact, what they did. So that's what the bead reconstruction looks like. And you can see that not only did they get the bead structure, but they also got a little bit of hints of the neighboring beads. And so not only did they know the bead structure, but they also know, we know, the connectivity between the beads. Okay. So just to show you what the polymer, the mucin 2 N-terminal polymer looks like, it looks like that. So here the beads are colored differently, which is nice, but it's actually not very informative. When we look at individual beads, all we can tell is the rotation and the translation between them, but we don't get any information about the mechanism of assembly of this protein until we start to look at molecules and not just at beads. So when we look at molecules, we see that each individual mucin molecule actually spans 2 beads. Just hold off on, that's not exactly true, but hold off for just a moment. Okay, in this picture, at least you can see them spanning 2 beads. So we have multiple domains within each mucin molecule that are separated by these cysteine-rich spacers. And I've called the short spacer a cradle because it cradles this D3 domain from the neighboring bead. And I've called this longer one a bridge because it allows the molecule to extend from one bead to another and position its D3 domain within the cradle of the neighboring bead. Okay, so what happens is that when you make a non-covalent dimer of the N-terminal region of mucin molecules, you keep these two D3 domains, which are indicated by these arrows, apart from one another. Now, I didn't tell you that yet, but these D3 domains have the cysteines that are responsible for making the disulfide bonds at the N-terminus of this protein. Okay, this is going to make a little bit more sense in the next couple slides, but for those of you who are still paying attention, I'd like you to try to help me throughout the rest of this talk because I have a big problem which I'm trying to solve. Now, I would like some sort of analogy to something in day-to-day life that follows the mechanism that I'm going to explain on this slide and the next one, and that's keeping something apart at an early stage to force it to interact with something else at a later stage. Now, the only analogy that I've been able to think of so far is not a visual analogy, it's just a sociological analogy. And that's that, some of you may know, right, that in Israel, people who grew up on a kibbutz tend not to marry any of the kids that grew up with them on the kibbutz, right? So if they're exposed to interactions, non-covalent interactions early in life, meaning in the endoplasmic reticulum in the case of this protein, then they tend not to form conjugal relations. So disulfide bonding reactions later in their lives, or in the secretory pathway, which means in the Golgi in this case, and they're in fact forced to interact with other people because of whatever biological mechanisms. I will not be able to use that analogy to help us get this paper published. So if anyone could think of a more graphical, more general analogy, please tell me afterwards, because I'd be very, very grateful. Okay, so where are we right now in the mechanism? We understand, right, that we have an individual mucin molecule that spans beads. This is all non-covalent, all the interactions between the orange and the blue. And the point of this non-covalent interaction is to keep the D3 domains separated from one another so that they do not make disulfide bonds with one another. Now we get to the next stage of assembly, and we're looking at, so all this is an analysis, by the way, of this single structure that we got from the polymer, but I'm just simply building upon it to explain the different parts of it. So here you see the orange and blue dimer that I just presented to you before. But now you see it in the context of the entire bead with the green and the purple dimer, non-covalent dimer, coming from the other side and forming the bead structure. And here we see that two D3s can actually form a disulfide bond. Okay? So this is the D3 from one kibbutznik, and this is the D3 from the other kibbutznik. They've met somebody from another kibbutz right now, and they've gotten married with a disulfide bond. Okay? So basically here we have the second step of assembly. This is the step that happens in the late-secretory pathway and ensures that the guys that were not that were originally associated with one another do not actually form disulfide bonds through their D3 domains. Okay, so there's one part of the story that I haven't told you about. One domain that's important. So the region that we looked at to make this low pH polymer actually contained everything up until this first orange domain. And this domain is called the cis-D1 domain. If we remove this domain, the N-terminal region of the mucin does not form a polymer. So this is absolutely essential for formation of a polymer by the N-terminal region of mucin 2. If you look in a two-bead reconstruction, so we simply expanded the box a little bit. You can actually see it in the one bead, but it's clearer here. You see that there's a linker that goes from a D3 domain, which is here, to the cis-D domain, which is the orange guy here. And you can sort of trace, you can't actually build the structure, but you can trace the density in going from the center of one bead into the next bead. And we determined the structure of the cis-D domain separately by X-ray crystallography to get higher resolution. And we saw there that there were a few exposed aromatic residues or hydrophobic residues on a loop. And this loop kind of sticks itself into the next bead. And this is probably helping to make stabilizing interactions that supports formation of the polymer. So in fact, each of the mucin molecules actually spans 3 entire beads. So this one starts off D1 here, D2 in one bead. It goes over through the bridge to the D3 in the middle bead, and then sends this PTS region, this highly glycosylated, slightly disordered, but not completely disordered region, into the third bead where it places the cis-D1 domain. Now, I'd just like to point out that this PTS region here is not the one that's labeled here. This one is 2,500 amino acids It's this tiny little guy here that's only 40 amino acids, and yet it's able to stretch 150 angstroms into the neighboring bead. So you can imagine, by extension, what this PTS region here is capable of doing. I should point out, however, that the short 40 amino acid PTS region doesn't seem to have too much repeat structure in it, but this is just a very small part of the 2,500 amino acids, and you can see that there is regularity to it. So if the entire point of this PTS region were just to be an unstructured glycoprotein, you wouldn't expect there to be such conservation of the repeats. So this is an open question about what this kind of structure is capable of doing, what it looks like, and where it's extending itself. Okay, so where we are so far is this is a summary figure that shows what we learned about the assembly mechanism of mucins. And I apologize if it looks horribly complicated. This is version number 27. You should have seen version number 1. But I'll just walk you through it slowly. So we have a protomer of the mucins. This is the N-terminal region here, up through the cis D1, which is where we solve the structure to. And I'm not showing it, but there are cysteine-rich spacers, right, the cradle and the bridge, that separate these D1, D2, and D3 domains. Then we have the PTS region and the cis D1. Then we have the very, very long PTS region that's not part of our structure. And then we have that CTCK domain that clicks in at the C-terminus and makes dimers. Okay, so what we have first is assembly of a non-covalent dimer between these N-terminal regions. And presumably, although we haven't shown it experimentally, the same dimer clicks in and forms the C-terminal covalent interaction by these CTCK domains. Now, at low pH, so when this structure is then exported from the endoplasmic reticulum and enters the Golgi apparatus, it experiences low pH, and we think that's the trigger for being able to assemble the beaded structure. Now, what does that beaded structure do? It places two separate dimers, non-covalent dimers, in appropriate position to gain their disulfide bonds between the two different dimers. Okay, now you can work this out at home if you ask me the slide, but if you don't want to, you can just trust me that if you break all non-covalent interactions present in this very complicated beaded structure, and you simply pull it out, leaving only the covalent interactions, what you're left with is the polymer that you wanted. So this entire mechanism, this entire complicated mechanism, exists to ensure that C and D are disulfide bonded here by their N-termini, but delta, which corresponds to D, the same molecule, is disulfide bonded to epsilon, which is part of E. So this is how to get a head-to-head, tail-to-tail polymer instead of a closed dimer through the same type of interactions. Okay, so you might think that because I'm at the summary mechanistic slide that we're done, but we're not, okay? And the reason why we're not is that mucins are not the only game in town. So it turns out that mucins are actually partial homologs of a protein called von Willebrand factor. So the N-terminal region that we've been studying in mucins is virtually identical, really very, very homologous to the N-terminal region of von Willebrand factor. And the C-terminal CTCK domain is identical in both cases as well, or very closely homologous in both cases as well. And in fact, the structure solved by Tim Springer that I mentioned was of von Willebrand factor and not mucin. Now, I already told you that mucins protect the epithelium, both in the gut and in the lungs. What's the job of von Willebrand factor? Von Willebrand factor is a molecule in the vasculature. It's produced by endothelial cells and platelets, and its job is to help in clotting. So it's doing a completely different job, but conceptually it's similar, right? It's preserving the integrity of the vasculature, just like mucins are preserving the integrity of the epithelial cell layers. Now, fortunately, von Willebrand factor can be run on a gel. It doesn't have these big PTS regions, and it's a bit smaller. So that's just to show you that, in fact, von Willebrand factor does run on a gel, and it makes polymers that you can actually see. Okay. So because of this homology with von Willebrand factor, the structure that we determined actually informs von Willebrand factor as well. Now, the history of study of von Willebrand factor structurally is much longer than the history of the study of mucins. It's been known longer, and it's a little bit easier to access, but not that easy. This is a picture taken from a paper by Peter Rosenthal. He actually even showed some of the images of von Willebrand factor in cells, but maybe didn't mention them by name. Mm-hmm. So this is a Weibel Palade body, which is the structure inside cells in which von Willebrand factor is packed before it's released into the the blood system. And Peter was able to do a low-resolution reconstruction from material in situ. And this is what that structure looks like. So the laboratory of Sadler at University of Washington, unfortunately, he's since passed away. So if anybody here knows him personally, please come tell me, because I really think he'd like this work. But I'd like to conduct a seance in order to transmit to him the information and get his opinion on it. Sorry, that's not very polite, but I really do wish that I could communicate with him and discuss this with him. Anyway, this is a low-resolution reconstruction of the amino-terminal region recombinantly in vitro of the completely analogous part of von Willebrand factor. So what you can see from these images is that von Willebrand does something that the mucins don't seem to do. That von Willebrand makes it makes polymers as well, but these polymers form helices, which there's no indication that the mucins actually do. So because of this tubular helix formation of the von Willebrand factor, the Weibel-Palade bodies have an order to them, a directionality to them. And if you look at them end on, you can see each of the individual circles of these tubules filling up this space. And this is very, very different from the type of packing we see from mucins. So for mucins, we have a whole bunch of different globular-like compartments surrounded probably by membranes, all smushed together into this larger compartment, this larger granule. But there doesn't seem to be any directionality to the packing of mucins within these compartments. So even though the two proteins are very, very, very homologous, they're doing something quite different in their supramolecular assembly. Now, because they didn't have atomic resolution information or residue resolution information, Sadler's lab and Peter as well weren't able to assign domains. But if we take their schematic model from their paper and we interpret it in the light of the mucin 2 bead structure, then we can understand the connectivity between the domains and von Willebrand factor. And moreover, even though we didn't have access to their original maps, their images were so nice that they presented in the paper that we were able to reconstruct a model for the von Willebrand factor tubule based on the structure of the mucin 2 amino-terminal region. And it really just slotted in there very, very nicely. All we had to do was play with we knew the dimensions of the tubule. All we had to do was just play with this rotation, and we were able to get a structure that looks very, very much like their low-resolution maps, inside and outside. I'll spare you the details, but this allows us to assign all the domains to explain why von Willebrand makes tubules and why mucins don't. We can map all of the von Willebrand disease mutations onto the tubule. So that's nice. Okay, what it also allows us to do is to compare the packing of von Willebrand factor and mucins. So this is the, shown in light gray here, it's a little bit hard to see. This is the tubule of von Willebrand, but shown in color are the pieces that are missing from the tubule structure and their connectivity. So in other words, the pieces that are connected by this these red lines and labeled 1 show where the rest of the protein, the entire C-terminal region of von Willebrand factor, would be packed. And that's the stuff that makes up the region around these tubules that you see here. So if you look at the mucin 2 polymer that we found, this beads on a string type polymer at low pH, you see that it's a completely different organization than the tubules, right? There's no wrapping around like this. You just have a kind of degenerate helix. But if you then attach the different portions that would be attached if we had the rest of the molecule present, you'll see that they actually come off on the same side of this extended polymer. And what I really liked about this is that they're staggered around this polymer with an angle of 98.5 degrees, which is very close to the angle between side chains in a helix, right? So completely different scale. But perhaps for similar reasons, this molecule has evolved to stagger this enormous PTS region that's coming out here, right, and making the rest of the molecule to avoid steric hindrance, just like we think that side chains of alpha helices are staggered to prevent steric clashes, or that's how it all worked out. It's not that. Yeah. Okay, right. So this is the down the polymer view of mucin, and this is along the side view of mucin, and you can see how even though they're such different structures, there are certain similarities between how the rest of the molecule wraps around the central tubule or polymer. Okay. So there's one thing that's very different between von Willebrand factor and mucins, and that's that von Willebrand factor lacks that cis-D domain, which, as I told you, is absolutely essential for making the mucin polymer. So as you can see here, no orange in the von Willebrand factor map. So what seems to be happening is that von Willebrand factor has additional stabilizing interactions that make the tubule form, and through formation of the tubule, it's able to guide disulfide bond formation properly. The mucins use the cis-D domain to help form a stable structure in the absence of tubule formation. But this cis-D domain also explains why mucins simply cannot form tubules. So in the von Willebrand factor tubule, the D3 domain is on the outside. Okay, it's labeled here. And in the mucins, this D3 domain is pointing up. And you can see that the cis-D domain is on the opposite side of the bead. So if mucins were to make a tubule, then the cis-D1 would be in the inside of the tubule, and that 2,500 amino acids of glycosylated PTS region would also be in the inside of the tubule, and that's just impossible. So mucin has evolved to keep all of the glycan parts outside and not to get caught up with the tubule formation and trying to stuff it into the inside. Without these restrictions, von Willebrand factor was able to make tubules. Okay, so I was really struck by Roger's talk and discussing how chromosomes are condensed, because it seems like mucins have a very similar type of primitive but highly functional packing solution. So unlike von Willebrand factor, which is assembled in a very orderly way into the tubules, our best guess right now is that even though the mucin structures do form polymers, the persistence length is not very great. If we look back at this original picture, we see that the N-terminal region makes strings, but it's not making really direct long polymers, right, that don't break. They're constantly breaking and changing directions. So if this actually exists in the situation of the real compartments here, then we expect all this material to basically be crammed in. We might have short stretches of polymer, but basically it's all just a condensed glycoprotein mass here. And therefore release is just going to be a kind of indiscriminate expansion of this material and not an unfolding or uncurling like you would have for von Willebrand factor. Okay, so that's where we are currently in our understanding of the system. So just to summarize, we've brought respiratory and gastrointestinal mucins and a blood hemostasis molecule all under the same umbrella by showing the similarities between intestinal mucins and respiratory mucins, and also jumping by homology to von Willebrand factor. And we've tried to analyze how quaternary structure guides successive disulfide cross-linking events, first in the endoplasmic reticulum and then in the Golgi apparatus, to assemble complex, highly functional protein networks that function outside cells. So we're doing our best to try to make mucus into a molecular science. So thanks very much for your attention. I'm happy to take questions. Sorry.
Thank you very much for that very interesting talk. U-shaped magnets, you keep the two poles apart so they don't stick to arrays of U-shaped magnets.
Another mechanism.
No, that's, you want an analogy.
Oh, the analogy. Oh my gosh, okay, all right.
Exactly that.
Sorry.
Wouldn't it? But that's where magnetism and disulfides break down as an analogy. But it's keeping the poles apart so they can only make a little picture with some.
Okay, all right, I'll think about that. Thank you.
Yeah, that was beautiful. I would say it was the sound of mucus. Any evidence for a pro-region cleavage in the assembly?
Okay, right, I didn't mention that. So von Willebrand factor, after it assembles into tubules, or at some point, I don't know when exactly, the D1 and D2 domains are clipped off. There is a recent paper on intestinal mucins that it's also thought to be cleaved off. And we see evidence that maybe we shouldn't get into too much, that, that human mucins are also cleaved, yes.
With so many cysteines, how many internal disulfide bonds you have, and is there an equilibrium between intermolecular and intramolecular disulfide bridges? And also, do you know if any cations are involved in in the interaction between molecules?
Okay, right, so there are more than 400 cysteines, so more than 200 disulfide bonds in the protein. And interestingly, if you mutate the ones that are involved in the intermolecular interactions, you do get some rearrangement, and you end up forming different disulfides intermolecularly. To what extent that happens during normal assembly, we don't know. As far as, you said cations involved? So calcium, the assembly and release is calcium regulated.
If I remember right, you said that the end cysteines would actually form disulfides in the endoplasmic reticulum?
That's what's thought, yeah.
But I thought the inside of a cell was highly reducing, and it's very rare to have a disulfide bond. Is that true?
So that's the cytosol, but in the endoplasmic reticulum is built for disulfide bond formation.
Okay.
So
that's in the cytosol.
Yeah,
yes.
yeah.
Okay, sorry.
So that's the preferred environment. It's rare to have a disulfide event in the Golgi.
Right. Okay.
Yeah.
And the other question was, this honeycomb thing. So you don't have a two-dimensional lattice, is that right? In the end, it looks more like a bunch of fibers, filaments, rather than a two-dimensional sort of lattice.
Right. So all we know is, I don't have a picture of it with me, but when you open up a mouse colon and you stain the mucins, it's very hard to preserve the absolute integrity of the mucin layer during fixation, but what you end up with is strands. So there's a directionality, and it's not a homogeneous network like this. So, yeah.
Okay.
Sorry, yeah. So my question is, usually with molecules from humans, there are a whole slew of genetic variants or knockouts with symptoms that tell you a bit more about the behavior. Are there any mucin variants or knockouts?
Okay, that's a great question. So there are a few variants. You would expect it to be associated with Crohn's disease and with colitis and all these other things. So mucin is like the deepest pit of GWAS studies you could ever possibly imagine. So last week we had this big biology meeting in Eilat, and there was someone who came to talk about the microbiome, and she showed a list of proteins that human variants were associated with differences in microbiome. And even though I knew it was hopeless, I'm scanning the list on on her slide, and of course mucin wasn't there. And I talked to her afterwards, and she's like, no, we do not deal with mucins. So von Willebrand factor, there are all these disease mutations of every single sort, well analyzed for however 80 years, and mucins are just not understood. So the sequence is too repetitive, too complex, and yeah.
What question? Oh no, actually, why don't you give it to him and then?
What sort of intestinal trauma would cause either the host microbiome or pathogens to start degrading the mucin? And what strategies do they have to break down these amazing protein arrays then? Right.
So I can't comment. I mean, I really, that's not my specialty. But what I can tell you is that if you get rid of an enzyme that makes disulfide bonds in the Golgi, that you completely change the stability of the mucus, and you also change the microbiome, and you start colonizing more with mucus eaters. Right? There are certain bacteria that are known to be mucus eaters, and certain bacteria that are known to be fiber eaters from the diet more. But that's like a whole big. Your question is like endless, right? There's a lot of.
Debbie, beautiful talk. I just want to point that mucin can be terrible in a pre-cancerous disease called pseudomyxoma peritonei, that the mucin is filling all the abdomen, and today there is no solution. What they do is just open and take out everything and wash with chemotherapy. So I think if your structure can lead to something that can degrade the mucin, it will help tremendously in the healthcare.
Okay, I'd like to hear more about that, so thanks.
Well, thank you very much, Deborah.
Okay, thanks.